So now I will welcome our speakers, Joakim Håkansson and Petter Björquist. Welcome.
Thank you very much. Then I will share my screen. Okay, so my name is Joakim Håkansson. I'm an associate professor in biomedicine and work as a researcher at RISE on the unit of biological function. And I will hold this presentation together with Petter Björquist at VERIGRAFT. You say hi as well?
Yeah. Hello, everyone. My name is Petter Björquist. I'm the CEO of the company VERIGRAFT. Here in Gothenburg, I have some 20 years plus experience from regenerative medicine and advanced therapies. First with another company that I know many of you know, Celotis, and since almost 10 years back, I'm with VERIGRAFT.
So we will have 4 parts of this presentation about the ATMPs, or tissue engineered products. First, we go through what kinds of different products there are in ATMPs. We will talk about the industrial perspective, why and how it's interesting for the industry to be a part of this field of drugs. We will show some results from common research that we have. And finally, we'll show how this now is tested on humans and how it will be tested in the future as well. So first here we see what kinds of different drug options there are. If you look at the right, you can see chemicals like aspirin, so on, biotech products like insulin and antibodies, transgenic proteins. We have vaccines, including the mRNA vaccines we have got now for the COVID, for example. These are not ATMP products, but then we go into the field here of gene therapy, cell therapy, and tissue therapies that we will talk about today. We also have medical devices that are not ATMPs, but if they are combined together with one of these other ATMP products, then it can be classified as an ATMP as well. And so to define the type of treatment or the modality, we can see that the chemicals here to the right are the smallest one, and then biotech products and vaccine, which can be up to 1 micrometer large. Then comes the gene therapy, also up to 1 micrometer, cell therapies that are between 10 and 50 micrometers, and tissue therapies, which are larger than, which we will talk about now. And then the largest ones are medical devices. One important aspect is also that there can be different sources of the therapy, the product. If you take the cells from the patient that will receive it back, it's called autologous to the left, as you see to the left. Or if you get it from another person or within the same species, from another human, it's called allogenic. But for the future, we also hope that we can take organs or parts of organs or starter material from organs from other species, and then it's called xenogenic therapies. So I will start then with giving an example of gene therapy, what that is. So, for example, we have the quite famous now CAR T cell therapy, which got the Nobel Prize in 2018. This is actually a combination of gene and cell therapy, but we go from a programmed gene that will express a defined protein in the body. We need to get it into the body, and then we use either viral vectors or a plasmid. And you can either inject that directly to the body, or you take out cells from the patient itself, add the gene in culture, and then give it back to the patient. In the case of CAR T cell therapy, then we take out purified lymphocytes, white blood cells, and with the help of gene therapy, we reprogram them, program them to express a specific protein that will recognize specific cancer cells, and then give it back to the patient so it can kill the cancer cells. Then we have instead cell therapy, which are somatic cell therapy, and it can be either cells that are substantially manipulated or if you take cells, you don't manipulate them, but you use them for a different essential function. So here it can be either autologous if you take it from the patient itself, or allogenic from a recipient, another donating patient. So if you go down here, either it's a substantial manipulation, then it goes back, and then it's an ATMP product, or if you make a substantial change for somehow, then it's classified as an ATMP product. If you just give them back to the patient, it's a normal transplantation, and then it's not classified as an ATMP product. And then we go into the to the subject of today, and that is tissue therapy that we will talk about. And then I give the word over to Petter.
Thank you. So after this very elegant introduction by Joakim, we would like to dig into a very specific area that is called tissue engineering, and the related product are called tissue engineered product. What we also will use as an example today from reality, that is that we will actually talk about organs. And why are organs so fantastic to talk about? And I hope that this very first slide will give you a little feeling of why this is so important. Because my company VERIGRAFT, we started a journey in 2014. We have worked side by side with RISE for many years. And after some 8-9 years of hard work and a lot of preclinical testing, we were ready to, for the first time, transplant a new organ to a patient. And that was in Spain last year. And I hope you can imagine how fantastic the feeling was when you called the surgeon and talked about what happened, and afterwards heard that everything went well and that the patient is really getting much better from a severe disease. But this was just as an introduction, so I would like to come back to this slide again a little later. So Joakim, have you given me the...
Yes.
I can't move the slides for some reason. There we are.
Yeah.
Very good. So what are we doing? We are actually working in the field of transplantation. And we hope to be able to help patients to beat their severe and chronic diseases. See, works very.
Isn't your that in that case I'll shift for you, but I have given the control to you.
It worked previously or earlier, but no, they
are
not.
I am shifting them.
Yeah. So let's then dig into this area of organs, and I give you a question now. Can we produce new organs to our patients? And I will try to answer that question. So in order to do that, I would like to give you a few personal reflections. So next one, Joakim. Autumn time in Gothenburg, it's very, very nice to show you a picture of a lobster. It's not because lobsters are fantastic animals. It's because this lobster illustrates exactly what we are going to talk about today. Namely, can we repair organs? And a lobster, if you break off a claw, you can see that the lobster just in days, 2 weeks, will start to regrow out a new claw. That is what I hope you can see in this illustration. And after only a few months, there is a new claw exactly as big and useful as the original claw. But how about us humans? Can we break off an arm or a leg and then see that leg or arm regrow into a new one? No. And that is because we are not equipped as lobsters with a very rapid and fully competent organ replacement machinery, one can say. So we need to help nature with that. And what can we do? So next one, Joakim. This is a cartoon you can see in papers nowadays. And I just want you to be a bit careful, because a new ear or a new outer ear, as this is showing, is not really science fiction today. This is just something that are sitting on your head and make maybe the your ear work a little better, but you can't go from deaf to hearing by having this right one. So the last one on the bottom here is, I think most of you know that this is a bioprinter, and you can see heart under the bioprinter. And is this reality today? And the answer is a clear no. We cannot bioprint the heart today. At least we cannot bioprint a heart that we can transplant. I'm a strong believer of bioprinting, but this is many years in the future. So bioprinting, yes, but that will happen for organs like a heart decades out in the future. So what can we do today? We are actually not creating organs. We are making them useful instead. So we call that for personalization, and that's what I would like to show you now. So next one, Joakim. The problem we are solving is the following. A lot of organs are transplanted today. So hearts and livers and kidneys are transplanted to us should we need a new one of these organs. But importantly, there is an extremely high price coming with the transplantation. And that is that you need to live on immunosuppressive agents, which means that you basically need to live without an immune system for the rest of your life. What happens otherwise? Yes, the organ will be kicked out by the immune system. So living without an immune system is really a high price because of two reasons. First of all, you should avoid big crowds, not go by bus, cinemas, etc., because you can die of a simple infection. Second one is that the drugs that are used, they are efficient, but they are connected to probably the most severe side effects we know today, increased risk of cancer, etc., etc. So the price is high, but of course, if we transplant the heart, we are paying that price because we will die otherwise. But a lot of other organs will not be transplanted because the price of living in the situation I described is higher than the benefits of receiving this new tissue. So next one, Joakim. Therefore, we, yes, transplant vital organs, but no, Joakim, next one, we don't transplant the non-vital organ. But does it have to be in this way? I think the answer is no. Next one. So there is, of course, something needed. And my company, we have a patent-protected technology that basically are solving this problem by changing the identity of a donated tissue. So next one. And next one again. So I would like to exemplify today's lecture. Data that Joakim is always later on will show is about vascular grafts, so vessels. So this is a blood vessel. This is a donor. The guy you see here, he donates or she donates a blood vessel. We are in the process taking off everything that gives an identity to this tissue. That is cells and DNA for the most. So next one. We then need to really understand that taking away cells from an organ, we don't end up in nothing or something completely different. If you take away cells from an organ, you will have basically the same for your eyes, you're still there. And that is because behind cells in an organ or a tissue will always be a scaffold. So this will be referred to as a scaffold going forward. The scaffold consists of protein and is responsible for the three-dimensional structure. This is important because we can do something with this scaffold. But please note that such scaffolds only consisting of proteins are easy to freeze down for a long time. So this structure we can put in the freezer for many years. Next one, please. Then we need to change the identity. And that is something that other scientists have done for decades. the most obvious thing to do is to bring up a new population of cells from the patient and add them. So typically in many projects, one has used stem cells from the bone marrow as one example. You take out the bone marrow, you identify the cells there you need, the mesenchymal stem cells, you isolate them, you put them in culture, you expand these cells for weeks, months, up to many times 4 to 6 months before you have enough cells that can be given back. We are not doing anything of that. We are instead harvesting a small sample of blood, and as illustrated here, we put the blood together with the scaffold. We are doing this in a bioreactor. We are adding some enhancing factors here, but we are not isolating anything. We are not culturing any cells. And this is very important because this process, next one, Joakim, ends up with a personalized organ. And in comparison to all our peers and other groups, this is done in as little time as 10 days. So compared to up to half a year, or at least many months. And in 10 days, we have a new identity of the organ, and we have something that is ready to be transplanted. So next one, Joakim. As you see here, this new identity will give us the possibility to transplant this tissue to the donor of the blood, being the patient. Change the identity, we have something new that can be transplanted. And the important conclusion of all is that this can be done without any use of immunosuppressive agents. Next slide. Just to sum up, this is then the most biological way of doing this, because when it comes to blood vessels, there are multiple other solutions either on the market or in development, but all of them that we know are doing non-biological approaches like synthetic grafts or semi-synthetic grafts. So this is the most biological way to work, and that is something that Joakim is going to answer now.
Yes, so I'm going to present some results from our common research. And we are working within the regenerative medicine field in several different areas, actually. We are working with veins and arteries that we will show today. We are working also on arthritis with cell therapy and tissue-engineered urethra and uterus. And we are several partners working on this together. So it's RISE and VERIGRAFT, also Sahlgrenska University Hospital, Roset, which is a GMP production company in Lithuania, Tata Biocenter from Gothenburg, YSDS, which is a logistics company, and also Gothenburg University. And we have had funding from different sources, also from Eurostars, from CAMP, which is the Center for ATMPs in Sweden, and Vinnova. We'll start to talk a little bit about the clinical indications. So one indication is that you can have a problem with the veins, with the valves in the veins. Petter will talk about that a little bit more later. We also have dialysis patients who need to be sticking themselves into the veins several times for dialysis, and then they need an arteriovenous fistula. And then there are people who have myocardial infarction. For them, there needs to be bypass surgery on the heart. And we also have people who have problem with the blood vessels in the lower limbs. The indication is called peripheral artery disease, where they need long arteries with small diameter. So this is sort of our main project plan. We have the process as Petter described, where we take out native arteries, decellularize them, and recondition with the patient's own blood. In this case, when we do animal studies, it's the recipient animal that we take. So the first project is for veins, and I can also show here that there needs to be very carefully analyzed all these parameters. So during this production of the personalized blood vessel, we do histology, we do immunohistology for different markers. We look with scanning electron microscopy and also pull and push in the organ with biomechanics. During the study, we examine the blood vessels with ultrasound, and we also do X-ray with angiography, so we can see that the graft that is transplanted is open. We, after the experiment is done, we take out the cells, do FACS sorting and RNA sequencing, so we can see that the cells that are in the graft, when the experiment is done for up to 1 year, is actually have the same function and gene expression as the native blood vessels. During this time, we also take blood samples, do FACS sorting, and look at the immune cells to see that we don't have any immune reaction. So the first project I will talk about is veins. So then we used a model on pigs to transplant the part of vena cava. And here is what the process looks like. First, here we can see the native, a picture of the native tissue, and all the purple dots, they are nuclei, cell nuclei. After the decellularization here to the right, we can see there are no nuclei left, but the pink is representing the extracellular matrix that we have, as Petter talked about. To the left here, you can see the bioreactor, how the blood vessel up here in A is white after the decellularization. Below in D, you can see a larger picture of it. And after the reconditioning with the blood, you can see that it's pink. It's very limited amount of cells there, but we have a biolayer of blood components that you can see here also in the middle section, the gray section here. Here we are looking actually at the lumen, the inside of the blood vessel. There's supposed to be a layer there of endothelial cells. So here in the native tissue, you can see here that we have cells there, a little bit hard to see, but in the decellularized picture, you can see that the surface there is really flat. There are no cells left. And after reconditioning, we have a surface with the blood components there. If we measure the DNA, we can see that in the native tissue, we have a lot of DNA. Almost all is gone after the decellularization. And after the recondition, it's elevated some, because there are some cells there, even though it's not so many. When we do this surgery, we work together with a blood vessel surgeon from Sahlgrenska University Hospital. And so here is the vena cava vein. We have the kidneys to the left and the dividing to the legs to the right. In the B picture, you can see that we have inserted the reconditioned vein part. We have a couple of metal clips here, so we can see it on X-ray to the right. So in the first study we did, after 5 weeks, we could see that all the blood vessels were open and nice. And to the right, lower right, we can see that the blood vessel had the surface that we wanted. There are no coagulations or trombones there at all. If we look at the tissue, what it looks like with the cells. So to the left here, you can see the native. It's the same for all 3 images. But what we want to see here is the personal tissue engineer vein, the PTEV. Already after 3 days, we can see that we have some cells coming into the tissue when it's inside the animal. After 2 weeks, it's almost the same amount of cells as in the native tissue. And after 5 weeks, we cannot see the difference of the number of cells there. So we have a really good recellularization of the graft in the animal. Here we look again at the luminal surface where we're supposed to have these important endothelial cells. And if you look to the left, the endothelial cells are marked in red with a specific marker. In the native tissue, you can see here that we have a thin layer of endothelial cells. And in the right, we can see here in the graph there that after 2 weeks, we have signs that we have starting to complete this cell layer. And in the L picture, we can see that we have a continuous layer of these endothelial cells. Below here, you can also see that the morphology is what it's supposed to be. The cells should be flat and in one layer. With these promising results from 5-week study, we wanted to make a new study so we could actually in the end continue to humans. And then we started, we did a study in mini pigs. and did the surgery for transplanted and held them for 1 year. So the images to the left is exactly the same as with the 5-week study. All the blood vessels were open after 1 year, and they had no coagulation or thrombosis. If you looked at the images to the right, you can see that we have the native tissue full of cells. After decellularization, there is nothing. Recellularization, there we don't have any cells in the tissue at all. But after 1 year in vivo, we can see that the graft has the same morphology as the native tissue. We can also, if we look more closely to the endothelial cells, we see that we have a nice coverage of the lumen, protecting from to the bloods. And one other important thing is that if we look in the blood vessel tissue, in the blood vessel wall, it needs to be revascularized with small capillaries to support the cells surrounding here with oxygen and nutrients. We have also done this gene expression analysis on the different cells. And the important thing with this graph is that for this pair of staples, one staple here is for the graft cells and one staple is for the native cells. You can see for all these marker genes that we have here, they are more or less following each other. So we have the same markers in the cells after 1 year as in native cells. And the results are now included in the regulatory documentation for an ongoing clinical study that Petter will mention a little bit later. So we go back then to this project plan again, and we want to proceed with arteries because, for example, in myocardial infarction patients, we would like to use arteries instead of veins. So then we use a sheep model. And in this case, we have also crossed the xeno barrier. So we are taking donor material from both pigs, sheep, and cow, and done VERIGRAFT process and transplanted them to sheep. The first study was for 4 months. Here we can see that the reconditioned artery is transplanted to the carotis artery on the throat. And after 4 months, we could see that the ultrasound, we had good flow. And we can also see here on X-ray between the arrows here that all the blood vessels were open. It's the same histology here. You can see that after the decellularization, we have no cells. But after 4 months, in this case, the morphology, histology is the same as with the native blood vessels. Here is again an image of these endothelial cells, also in arteries, that it works fine. We have the endothelial cells with electron microscopy. We could see that the whole surface is covered. And also in these arteries, which have a little bit thicker blood vessel wall, we can see that they are revascularized with capillaries in there to support the cells with oxygen. This has worked fine for both the material from donor pigs, sheep, and cow. And we have done a 1-year study for all of them, and all the blood vessels have been open so far. And we are now analyzing the molecular biology analysis from these studies. So I will give now the word back to Petter to show how this is used then in patients.
Thank you. We can see Joakim, if I can move
yes
the thing myself. So what about again all these years of hard work on the bench and in all these kinds of animal models? We also had the final target of receiving the real model, and that is the human being. So our blood can be either
Petter, I think you have a problem with your internet. Could that be so? We can't hear you now, I'm afraid. Are you still with us? Or okay.
Can you hear me again?
Yes. Now you're back.
I'm back. Very good. You
So
probably lost me.
Yes, we didn't hear you the last 30 seconds.
Okay. So Joakim, I just said that I can't move the slides.
Okay.
So if again, yes. So as Joakim already said, in this particular vein, we have a system that is meant to regulate the blood flow up and down from the feet. I think most of the oxygen-rich blood from the heart out to the extremities, leaving the oxygen, and then leading the blood back to the heart again. And that is done in this particular vein. But the problem is when the muscle pump is pushing up the blood to the heart, gravity tries to get the blood down to the feet again. Then we have these elegant valves that are letting the blood go up, but disallowing the blood to go down again to the feet. Unfortunately, more than 2 million patients only in the US, to give you one example, are having insufficient valves. Insufficient means failing, non-functional, and can look like this. The blood cannot be trapped in the valves any longer and are constantly going down to the feet. That is causing a problem called chronic venous insufficiency. And these problems are starting with swelling of the leg, a lot of pain in the leg, but unfortunately, gradually, leg ulcers are formed. Next one, Joakim. And that can actually look like this. So it's really a terrible disease, and the patients are not able to live a normal life any longer. Most patients are out of work. They are on early retirement. They have a quality of life that is very low. They cannot go to the beach. They cannot ski. They cannot do anything else that us normal people can do. So this is obviously a huge unmet medical need. And therefore, we started the journey here. Next one, Joakim. So we developed our first product, and that is what you see here. The product is basically a 4 to 6 centimeter long segment of these veins, looking like this. And it's nothing strange. This is something that Mother Nature has created. But if you have followed our presentation today, we have changed the identity and personalized these grafts. So now we can transplant without the use of any immunosuppressive agents. So this product is ready to be transplanted. Next one, Joakim. And next one again. In reality, this product looks like this. This is a lab simulation of what I just told you. I think you can see the valves working when you push the liquid in normal life, blood from below and up. And the valves are closing when they go down again. Next one, Joakim. So we are now doing this in a clinical trial setting. We decided to do this in Spain. And for those that are interested in why we are doing this in Spain and not in Sweden, when we started to prepare for this clinical trial some 4 to 5 years ago, there was not really possible for us to do this in Sweden because Sweden was not really ready. So ready here means that our authorities were not really educated. There was nowhere where we could produce the material in a clinical grade setting, and there was a lot of other things missing. Now I think it would have been different, and as at least some of you know, there is something going on in Gothenburg called CCRM Nordic, and that will solve exactly this problem. But we did this in Spain, so we have a trial called First in Man trial, or a phase 1/2 trial, consisting of 15 patients with severe chronic venous insufficiency. So the question is then how this is working, and luckily the data that is coming in is very positive. So the patients that really had a lot of problems are having less problems. This is a safety trial, so most focus is on safety, meaning that we look for side effects or unwanted effects that can be thrombosis, that could be infection in the grafts, that can be mechanical failure in the graft, and that could of course be the main thing here, rejection, should our technology not work. But luckily enough, with about half of the patients now being treated, we haven't seen anything, no single sign of any of these 4 major worries that we had before the trial. And since these are patients, we cannot avoid to see that also symptoms are getting better and better. So a little early to conclude, but everything looks very positive. And now we get the payback for our years of hard fighting. So next one, Joakim. We are a company, and I guess some of you at least can be interested in how a company is handling a project like this. So our business model is very simple. VERIGRAFT will in the future be a producer of these personalized tissue engineered grafts, at least in the westernized countries, Europe and North America. In the rest of the world, we today think that we will out-license this technology to a partner. Next one, Joakim. Something that is very, very interesting and very, very important in our field, that is the so-called reimbursement model. And you do that by looking at health economy. You make some analysis on how much you can save in terms of costs for the hospital, cost for the societies, cost for a country like Sweden. And on the good side, then we have worked with 2 different experts here, and the reimbursement model is always turning out to be very positive, meaning that we can really in the future sell this, save money for the patient. If we can save pain and suffering from the patients, we can save money for the societies, and we can also earn some monies ourselves as a company. Next slide, Joakim. Mm-hmm. So we have talked a lot now about personalized veins. I just have to mention that we have other products in pipeline. Joakim has mentioned that also. We have personalized arteries. They are in large animal trials. We hope that we will be able to start a clinical trial in this field in approximately 1 to 2 years. We are also personalizing nerves, heart valves, and urethras. So this technology has an immense set of opportunities for the future. Next one. Now I come to something that is very important, and I hope that you at RISE will find interesting. As you have seen, we are basing our product today out of human donated material. But there will always be a question, is there enough human material to supply a huge medical need? And the answer is that we don't know that really, but it may be a no to that question. So we are working on 2 other very interesting alternatives. Next one, Joakim. Tomorrow, I say here, we think that instead of having humans as donor, we will have animals as donors. So animals will be the ones supplying us with the scaffold. So for arteries, for example, many of the traditional farming animals you have seen Joakim talk about, cows, sheep, and pigs, are having arteries quite similar to our arteries. So we can decellularize those arteries and we can repopulate with material from the patient. This is, of course, increasing our access to starting material quite a bit. But we are not ready there. We have a third one that will become a little further in the future, and that is to use printers instead of either humans or animals. So the day we learn how to print a scaffold with high perfection and at high scale, we will have a machine standing there and supply our material, and we can personalize the scaffold produced by a printer. And I hope you can agree that this would be absolutely fantastic. We are independent of any donors, and we have an unlimited supply of material. And maybe it's not too far away in the future, but some 3 to 5 years at least before we can see this in the clinic. So having said that, we are coming to the end. So next slide, Joakim. I would like to show you the lobster again, because it's always nice to circle back to what you start to say. Lobster is a fantastic animal. We human beings cannot produce a new arm as the lobster can do, but we can help Mother Nature with a lot of other things, and we will be able to personalize organs, and we will be able to help patients in need of new spare parts. And let's see, next click, Joakim. If the lobster is regenerating, you can see here, and we also hope to regenerate. And by that, I would like to, on Joakim's and my behalf, thank you for listening to this. We hope that you have got a bit of a summary of something interesting ongoing in Gothenburg. I think you have got the glimpse of an absolutely fantastic collaboration between the industry and the research institute that you all are working on. And without RISE, I could say this journey would have not been possible to make. So again, thank you for the collaboration, and thank you for listening.
Thank you, Petter. So let's see if we have any questions. So how long do you expect your personalized blood vessels to function after transplantation? Is it Joakim?
Yeah, I can answer that. We expect them to last for the whole life. That is the goal.
Fabulous. Let's see. And what gains do you anticipate for the society when these products once reach the market?
I take that one then. So, I mean, the whole idea here is to have benefit for someone. And I think we have a lot of stakeholders in a project like this, and a product developed of this kind. So the first and maybe the most important stakeholder here, that is, of course, the patient. So the patient will receive a new graft, and in the situation of a new blood vessel, as we told you about here, I can tell you that the patients we are treating in Spain, they are patients that are very little mobile. They are sitting at home. They have a lot of pain. They can't do a lot of the fun things that other people are doing. So they are, I think I mentioned that, typically with a very low quality of life. They have also very hard to work. They are very often on sick leave or even early retirement. So on the patient side, we have a lot of benefits. But when it comes to the societies, there are, of course, a lot of other benefits indirectly, that is saving costs and other things. And that is, for example, that in a country like Sweden, the calculation that you can find is that one person that are forced off work, but that are allowed to go back to work, will be a benefit of about 1 million Swedish crown per year. And as Joakim said, our grafts will be fully integrated and will last for the rest of the life. So our patients are very often even in the age of 40 or 50 even. So you can imagine how many years you can save 1 million crowns. If you look at the even higher level, there are so many other benefits for the society to have patients being healthy and working and exercising and whatever that these patients cannot be. So a lot of gain will be out of this. Thank you.
Thank you. We have a question from Jennifer Rosendahl, and she asks, do you have any other applications where your veins or arteries could be used?
As a company, you always have to look at opportunities that are not only sort of scientific opportunities or biological opportunities. So as a company, we have decided that this first vein product, and specifically a piece of a vein with valves, are the perfect start for this journey. And I think actually I didn't mention the most important reason, because there are no other solutions on the market. And a patient today with chronic venous insufficiency is only receiving symptomatic treatment, meaning that the patient can get help with swelling, that is surgical stockings, the pain, of course, painkillers, and the wounds are taken care of with a lot of wound care. However, of course, we have a lot of other veins, Jennifer, in our bodies, but we see that as very small indications, or rather a very little medical need. So therefore, we will jump over from veins to arteries with our second product, because arteries are containing some really terrible and diseases and some enormous medical need. To give you 3 examples, arteries can fail in the lower parts of the leg. That means that patients can have poor blood supply above the knee, is typically the description. So the lower part are not receiving oxygen via the arterial blood, and leading very often to amputation. So if we can avoid amputation and really a terrible life, that would be one of our next areas. Another one is called coronary artery bypass grafting, where you need a new artery that is supplying any part of the heart with oxygen. So after a heart infarction, for example. So that will be another one on the arterial side. And the third one is an area where dialysis patients are needing a new artery to do dialysis, because when you are on lifelong dialysis, you are in need of a sort of a spare part that can be used for the dialysis process. So that is also a huge medical need. So those areas are all today partly targeted by synthetic grafts, but there is still a huge medical need. So we believe that the answer to your question is that we will only at this point target the groin region with our veins, but then we will try to supply or help people with all their problems on the arterial side.
Excellent. We have a few minutes left. So do you have what competitors exist around the world?
Yes, I partly mentioned that in my previous answer, but interestingly, the market for, or I should again start with veins. And basically there are nothing on the market. So that's an area without competition. But on the arterial side, there is in one way fierce competition, because we have some big dragons that are today selling on $1 billion market. And those are the companies that are selling synthetic grafts. And just a funny thing here is that the American company Gore, of course, producing Gore-Tex. And some of you may think that, well, they are doing jackets and shoes. They do. But Gore are using Gore-Tex as, for the single most important product for Gore is a blood vessel out of Gore-Tex. So that can be good to know when you read the newspaper about Gore and Gore-Tex, that blood vessels is really an area where they are dominating. So they are a big competitors. In reality, I think it's both bad and good with competition. So Gore will be hard competitors on that market, but we know that a fully biological solution will kick out Gore from at least part of their market. And when they see that, they may be so interested in this, so they invest or even buy VERIGRAFT. So that can be a nice sort of end of that story some time in the future.
Fabulous. So what do you see as the biggest obstacles or risks to the success of this concept on the way to commercial success?
That's another very good one. And I mean, Joakim has talked about some of the obstacles, and that is to see that this is working in a biological system. So all the animal studies that we have done and other studies that RISE has done with the material is taking off the early obstacles. Then we have, of course, the clinical journey. We need to prove ourselves in the clinic before we can go to the market. But I would like to lift up a huge risk, and that is that we ran out of interested investors for this, because I guess that you understand that this is something very expensive to develop, and we need someone to finance it. And today we have attracted a global set of investors from Europe, from Asia, and from North America that have invested money in VERIGRAFT, but more money is needed. So I would say that maybe the hardest nut to crack is to finance this going forward, because when you go into the little larger clinical trials that you have to do just before you go to the market, those are very expensive. So if you have any contacts in this audience to people out in the world that would like to invest in a company like this, you are more than welcome to help us solving this problem.
Thank you, Petter. We have a question from Maria Danasica. Would this be accessible to most people, or would it be very expensive and exclusive?
That's a very good question. And first of all, I have to answer that is dependent on where in the world you live, because in some parts of the world, the government will take care of this. I think Sweden is a good example. I mean, should we be able to prove that we will cure a severely ill patients that are off work and can bring that patient back to work, I'm absolutely sure that the Swedish taxpayers will understand this and save money by investing in this treatment and paying for us to do this. In other parts of the world, like in the US, it's mostly insurance companies that are paying for medical care. So there it's up to the insurance companies. But again, if we prove that we get the person back to work from being off work, it's easy to see that the insurance companies are going to pay for this. In yet another parts of the world, unfortunately, maybe we can say as Swedes, there are private healthcare, and then it's up to each person to pay or not for this. I think you have many countries with that system. And then I guess it's about if you can pay or not for this. But I think in general, if we prove that we will cure patients, I think this will be really widely and globally used.
Great. So do we have any final questions from the audience before we finish this webinar? Well, right. So I want to thank you, Petter, and Joakim also. Thank you very much for very, very nice presentations. And thank you, the audience. Thank you all for listening. And take care of yourselves.